हेलो,
मैडम। हाँ बोलिए मैडम।
ठीक है, मैं शेयर कर देती हूँ। आप डॉक्टर जैसे ही मुझे नेक्स्ट करने के लिए बोलेंगे, आई विल चेंज।
ओके, ठीक है, तो स्टार्ट करें मैडम। हेलो अरुण मैम।
आप अपनी तरफ से स्टार्ट कर दीजिए, मैं शेयर करती हूँ। मेरी स्क्रीन विजिबल है? ठीक है सर, आप मुझे नेक्स्ट बोलेंगे, मैं नेक्स्ट करती हूँ।
ठीक है, ठीक, ओके, नो प्रॉब्लम. Okay, good afternoon, all the audience, respected doctors and the colleagues, my dear friends. So we are going to discuss about novel G protein vaccine that is groundbreaking for the anti-rabies vaccine schedule. That is 3-dose vaccine, and that is said that it is ultimate key for the compliance. Next.
टेक सपोर्ट लाइव है ना? हेलो, टेक सपोर्ट लाइव है ना? हेलो,
Next slide. Next slide.
हाँ लाइव है मैडम।
So we'll start with our presentation about first we will see a few data, epidemiological data regarding rabies. It was and it has been a very headache like situation. It has been public health challenge for the India. Looking at the data of death by rabies globally, roughly 59,000 human deaths occur annually. Out of 59,000, 20,000 human deaths per year from the rabies is only from India. So if you count, it is roughly every 9 minutes there is one death from rabies. Next. So coming to the current management, we all know that we do have precise guideline regarding current management. First of all, we will do first aid, that is local wound treatment. Then coming to the immunization, it can be active as well as passive. Active by giving vaccine and passive by giving RIG. So for that previously, yes, for that right now we are using 5-dose schedule. That is day 0, day 3, day 7, day 14, and day 28. We are using this intramuscular regime. Next. Next slide. But it was and it is current challenges for the post-exposure prophylaxis for the anti-rabies vaccine. That is non-compliance. Current rabies vaccines are cell culture vaccines. Inactivated vaccines were greater than 50 years old. Technology inconsistency was the main thing. Stability issues were there. Availability was very troublesome, and adverse events were also there. Next. Now coming to the compliance part, that is very important part in the management of anti-rabies vaccine. And personally, I feel that compliance is a very important issue regarding anti-rabies vaccine. So there are three categories for this compliance. We will call compliant when the patient who has received full course of anti-rabies vaccine that is on schedule in recommended dates. Then coming to the next terminology, that is delayed compliant. The person who has completed the scheduled dose of ARV within 60 days, but deviated from the schedule regime. And the third one is dropout, or you can call it non-compliant. The person who had taken at least one dose of ARV after animal bite, but not completed all the scheduled doses. Next. So coming to the next slide, it has been documented that previous slide. It has been documented that non-compliance to 5 doses proven as the cause of death. The reason of death may be due to lack of administration of the rabies immunoglobulin or poor compliance to the vaccination. It can be either of it, or it can be both. Next. Next slide. We do have very reliable data regarding previous slide. We do have very reliable data regarding compliance of the this post-exposure prophylaxis. It said that the study was conducted at Safdarjung Hospital, New Delhi, where 360 patients were enrolled. It was pre-designed, pre-tested structured questionnaire was administered to the interview technique. And after this study, it was concluded that 53% were non-compliant to full course of ARV. That can be the main reason of this type of mortality in rabies. Next. There is another study showing that compliance is the very important issue regarding anti-rabies vaccine, particularly 5-dose post-exposure prophylaxis. So compliance rate for, in these studies, it was said that compliance rate for intramuscular rabies vaccine was 60%. Next. So coming to the constant of compliance, what are the causes of this non-compliant thing that were analyzed? 11% patients said that it interferes with the school timing. Another 11% said that distance from the hospital was the issue. 13% had some cost issues. 18% had some forgotten dates like issues, and 47% had the issue of loss of wages. That is what they were reluctant to take regular anti-rabies vaccine. Next. Okay, now this slide is showing the evolution process of anti-rabies vaccine. You all have heard about it, or sometimes you may have given or sent patient for this 14 injection regime. So it started with 1885, 31 injections were given. That was non-standardized dry preparation. That was followed by inactivated vaccine roughly in early century of 19th early years of 19th century. Minimal neural content and liquid preparation was there. 14 injections were given, 0.5 ml of each daily. Then the groundbreaking vaccine, that was cell culture vaccine, was inactivated vaccine, was discovered in 1976. That we are using nowadays, that is 5 injection schedule, that is standardized immunogenicity and lyophilized. Next. Non-compliance led to evolution of treatment regime. Obviously, if we have to take 31 doses, non-compliance is the issue. So now, because of non-compliance, the evolution of anti-rabies vaccine started from 31 doses to 5 doses, and still it goes on. Next. So the problem is non-compliance. The problem is multiple dose. The problem is non-compliance and the death rate. So we do have the solution. Next. Novel 3-dose recombinant anti-rabies G protein vaccine. That is first vaccine with a 3-dose intramuscular 0-3-7 days. It is prepared from VLP platform technology. That is some technical term. First protein subunit vaccine. It is in views of compliance. It is better compliance due to easy to follow regime and less visits. Convenient, ready to use, no reconstitution needed. So these are the advantages of this novel 3-dose recombinant rabies G protein vaccine. Next. Next. So it is this slide is showing what is VLP technology. So basically it is some technical terms. The native virus is G protein is difficult to stabilize, and G protein is isolation is purified, stabilized by the Cadila Pharmaceuticals. Cadila Pharmaceutical is specialized in isolation as well as stabilization of this G protein from the native virus. Then VLP, that is virus-like particles, are protein complexes exist as nanoparticle, and this is how it is prepared. It is very capable of inducing a strong humoral as well as a cellular immune response. Initially, I was also not able to believe that it can produce this humoral and cellular immune response, but I used it in my lots of patients, and it is producing humoral as well as cellular immune response. Next. Okay. We as doctors, we need some strong data regarding this cellular as well as humoral immunity or immune response. So this is the study, clinical study phase 3. The study was safety and immunogenicity of this 3-dose vaccine, recombinant nanoparticle G vaccine in simulated post-exposure immunization. It was randomized, comparator control, multicentric phase 3 study. Next. Next slide. Okay. I would like to show some throw in this phase 3 study. It was all the clinical trials are done in very stringent conditions, and this study was also done to evaluate the immunogenicity as well as safety of this vaccine post-exposure prophylaxis. It was randomized, comparative, controlled, and multicentric studies. Total 800 subjects were enrolled, and randomization was done on the 2-1 basis. That means 267 subjects were randomized to that 5-dose regime, and 533 subjects were randomized to the rabies, that is 3-dose G protein vaccine. Next. Next slide.
Next slide.
Okay, now coming to the results of this study, that was phase 3 study. It was compared that efficacy and safety both were compared in all the clinical trials. It is compared that efficacy as well as safety are compared. So after the phase 3 study, it was found that non-inferiority for the seroprotection primary endpoint at the day of 14 as well as 42 were met. So it was very similar to that 5-dose regime. It is shown in efficacy chart. In compared to test and comparator are identical in the seroprotection rate, both in 14 as well as 42 days. It is if I throw some lights on the charts. In first chart of efficacy, it is showing seroprotection on the day of 14. It is almost similar, 99.27 and 24% in rabies. 97.72% was by that 5-dose regime. So our was even better, but statistically both were non-inferior. Similarly, coming to the adverse event, it was statistically significant lower in compared to this 5-dose regime. Local adverse event or systemic adverse event, every adverse event were very lower in compared to 5-dose regime. So it is efficacious, but very safe. Next slide.
Ma'am, please next slide.
Next slide, please. Okay, so coming to the conclusion of this phase 3 study, novel 3-dose vaccine was non-inferior to that our 5 days 5 days regime, less adverse event compared to 5 days regime, and novel 3-dose vaccine was found to be safe, immunogenic, and was comparable to the 5-dose WHO pre-qualified vaccine. So it is equally efficacious but more safe. Next. Okay, the Cadila Pharmaceutical Company has got this prestigious awards for this innovation of this product. Next. I would like to thank the Cadila Pharmaceutical Company, the management, the technical staff, the medical representatives, and August audience. Thank you so much.
Thank you, sir, for being part of this webinar. I will ask Shehzad to ask few of the questions which are there in the comment section so that you can answer, sir. Just give us some time.
Yeah. Sure.
Sir, one very common question which has been asked from like across India.
Okay.
What are the bites? Like only dog is responsible for rabies, or any other uh, uh, animal is there for which are also responsible for rabies?
Sorry, I didn't get your question.
So the question is like rabies is caused by only dog bites, or for other animal bites also it is common?
Can be more spread of rabies by dog bite is more common in compared to other animals, but 96% of the dog bites are responsible, but it can be spread by another animals as well.
Okay, sir. And one more question, sir. Like if any dog bite is there, then what is the initial immediate measures should be like a patient should take?
Okay. First of all, it should be the wound should be thoroughly washed and flushed with the soap and water for roughly 10 to 15 minutes. The wound is not to be ligated. It should not be. It should be kept open and flushed with the thoroughly with the water. Wound should be cleaned thoroughly with the 70% of the alcohol, ethanol, or spirit, or povidone iodine, any of the antiseptic available at the site of this first aid is given. And after this wound care, as soon as possible, the person should reach to the healthcare facility and should take active as well as passive immunization.
Okay, sir. Thank you so much, sir, for being part of this webinar. And thank you all the participants who waited and they have all our ears wherein listening to all your conversation. And it was a wonderful presentation by you, sir. Every point and everything was very clear. So thank you so much for being part of this
Thank you very much.
journey. Thank you so much. Yes, sir, we can conclude the session.
Yeah, it was nice experience for me as well as the team. I would like to thank Kerala as well as the whole technical as well as managerial staff. It was very fruitful. I hope it was very fruitful seminar or the webinar for all the
Yes, sir.
audience.
Yes, sir. Definitely. Yes, sir. Thank you so much, sir.
Thank you. It was pleasure. It was pleasure for me as well as to talk to you all. Thank you.
Thank you, sir.
Thank you.
Thank you. Good day, sir.
Same to you. Thank you.
Tech support, please conclude the session.